Hello everyone, Mrs. Hanson here on a nice Tuesday morning, getting ready to start chapter 21, the first of our lessons, investigating amino acids, proteins, and enzymes. So again, a next biochemistry chapter featuring molecules related to amino acids, which are the building blocks of proteins. And then we'll look at enzymes, which we often hear as nature's catalysts. Let me share our learning goals. In our chapter, we want to be able to identify the general structural features of an amino acid. We'll describe the acid-base property of amino acids. We'll draw simple peptides, which are just chains of amino acids together, labeling the nitrogen terminal end and the carbon terminal end amino acids. We'll give examples of biologically active peptides. We'll discuss the characteristics of a primary, secondary, tertiary, or a quaternary structure of proteins. You'll describe the features of a fibrous protein, like an alpha keratin and a collagen molecule. You'll be able to describe the features of globular proteins, such as hemoglobin and myoglobin. You'll draw the products of a protein hydrolysis, adding water. What happens when a protein becomes denatured? What are the main features of enzymes, and how do we classify enzymes into one of 6 categories? I'd like us to discuss 3 types of enzyme inhibitors and discuss the use of enzymes to diagnose and treat diseases. So let's begin in investigating proteins. We said that there are 4 major groups of molecules. We started with lipids. We just completed carbohydrates. In this current chapter, our focus is on proteins. And in our next and last chapter together in our semester, we'll focus on nucleic acids, DNA and RNA molecules. Of all of those 4 major groups of biomolecules, proteins have the widest array of functions. Remember, lipids had a wide variety of structures. Proteins have a huge, just an overwhelmingly large number of functions. For example, one category of proteins are the keratin and collagen molecules. These are long, insoluble fibers that give strength. Think of your hair, horns, hooves, fingernails. These are all keratin molecules. Collagen is found in bone, connective tissue, tendons, and cartilage. So insoluble fiber, you can see these molecules give strength to these structures. Another area we often find proteins is embedded in cell membranes. These cellular proteins transport small organic molecules or ions across the cell membrane, making it selectively permeable. You can have passive or active transport at the site of those proteins. Another protein is insulin, hugely important in regulating blood glucose levels. Have you heard of hemoglobin? Of course, in our blood, it transports oxygen from the lungs to all of our body tissues. And enzymes, those nature's catalysts that help regulate the rate of a cellular reaction. So, with your notepad ready, let's begin. Proteins are biomolecules that contain many amide bonds formed by joining amino acids. So just to kind of bring an idea of where we're at in our notepad, just bringing this to a little bit of beginning area, we are just looking at proteins as biomolecules formed by joining those in a peptide bond. Let's begin right in this section by drawing in a general structure of an amino acid. Now notice we have what we'll label as an alpha carbon. This carbon has 4 attachments, just as every carbon does. 3 of the 4 attachments are always the same. We will learn that there are 20 naturally occurring amino acids. And so of the 20, 3 of the 4 bonds are going to be the same from that central carbon. and it's the fourth bond that makes the identity of the amino acid known. So of the 3 bonds, one of them will always lead to a hydrogen. A second will always lead to a carboxylic acid functional group. Carboxylic acid. The other will lead to an amine group, NH2. And it's this R group that gives the amino acid its identity. That fourth bond is unique in all 20 amino acids. That fourth bond leads to a functional group that gives the acid its identity. And I'm going to just use AA as amino acid, and that will be a nice shorthand way for all of us to refer to an amino acid. It gives the amino acid its identity, and we'll show you in a little bit by looking at a chart of all 20 naturally occurring amino acids. So a general structure, we have a central carbon. In 19 of the 20 naturally occurring amino acids, whoops, not 200, 19 of the 20, that carbon is chiral. It's just one of the 20 that central carbon is not chiral. It leads out to another hydrogen, making it achiral. And we'll draw that in just a moment. So we have a central carbon leading out in one direction, carboxylic acid. There's the acid part of the word amino acid. In the other direction, we have the amine group. Can you hear those 2 words? Amino acid. One of the 3 bonds is always to a hydrogen, and this is what's unique, giving the acid its identity, is that whatever R represents, that alkyl group for R, will have one of 20 possibilities of an amino acid. Now, amide bonds join groups of amino acids. So if I think about an amino acid where we have a terminal nitrogen, I'm going to just color code these so we can see how they're different from one another. The NC to the carbonyl, NC to the carbonyl. And I can show you that we have different amino acids kind of grouped together. And highlighted in red, you can see the amide bond. And I'm just kind of emphasizing by color coding those even deeper in red. Those amide bonds are connecting one amino acid to the next. Now, in organic chemistry, the amide bond, which is right here, we'll learn is going to just form from the splitting out of a water molecule, one of the hydrogens from the amine and an OH from the carboxylic acid split out. And when you remove one water molecule, it's known as a dehydration. Remove a water, one of the hydrogens here with an OH here, see the HOH? That sets up the connection then for the nitrogen from one amino acid to bond to the carbonyl group of the other amino acid. That bond that joins one amino acid to the next is known as an amide bond from the nitrogen group. You'll also hear it called a peptide bond. Amino acids, when they begin to join one to the next, one amino acid bonded to one other, will be known as a dipeptide. Dipeptides can go on to form polypeptides and then polypeptides, there we go, polypeptides go on to form true large structures called proteins. There. All right, so we've taken the time to draw out a general structure, and we've drawn a long chain. Notice that these little squiggly marks say this is going to go on and on and on and on, one amino acid to the next. The amino acid identity comes from whatever this R group is. There's one of 20 choices that can be, since there are 20 naturally occurring amino acids. Proteins account for about half of all the dry weight of our human body. And unlike lipids and carbohydrates, proteins are not readily stored, so they must be consumed in your diet every single day. The current recommended daily intake for adults is about 0.8 grams of protein per kilogram of body weight. And just to give you an idea, 1 kilogram is 2.2 pounds. To give you an idea of that equivalency. Children, of course, would need more because they're growing. And then dietary protein comes from sources such as eating meats or drinking milk. And here is just a nice visual of all the different types of proteins and their functions. So these kind of lists just right down in order on your notepad page. So you can see as I scroll down, this is where we're filling in now. Alrighty. And so, for example, hemoglobin. That's this molecule here, hemoglobin. We find hemoglobin molecules in our blood, and they are transporting oxygen from the lungs to all the body tissues. So their major function is transportation of the oxygen to our tissues. Next one says collagen. We mentioned that in our opening statement. Collagen is a type of fibrous protein, and it's found in connective tissue. It's very rigid. It's found in tendons and bones, cartilage, and in blood vessel walls, especially to give it strength. Ferritin. Ferritin is a protein that stores iron in liver. Do you hear that root word for iron? Ferrous and ferric. That's our Latin word for iron. You can hear that in the root word here of our protein. And oftentimes people will eat liver, and I am not a fan of eating liver, but some people do. And when you eat liver, it's very rich in iron. That's because iron is stored in our liver in a protein called ferritin. Two molecules you might have heard of in anatomy and physiology, actin and myosin. Those two proteins work as a team and controlling muscle contractions. Keratin. This is a fibrous protein in hair, in your fingernails, and in your skin. Myoglobin. That's a protein that stores oxygen in the tissues. And finally, insulin. Insulin is a protein that's synthesized in the pancreas to help control blood sugar levels. So people who have diabetes have a hard time regulating their sugar in their blood levels and require insulin sometimes to help control that. So an idea of all the different types of functions and varieties of protein molecules and what they're, just a brief look, because remember, this is a wide variety of molecules. So next, let's take a look at some general features, diving into the structures a little deeper. I know we've drawn a general structure. Let's get into the detail. Amino acids have two functional groups, and you can hear it in the name. An amino group, the amino group, of course, is NH2. And the carboxylic acid group, COOH. Carboxylic acid, COO, and the amine group, NH2. And we've had chapters uniquely devoted to each of those two functional groups. What makes an amino, think nitrogen, acid, think COO. What makes it unique is it has a basic and an acidic functional group all within the same structure. The amino we know is basic from the derivative of nitrogen. And the lone pair up here of electrons makes it an electron, or we'll call it a proton acceptor. We'll go with Bronsted-Lowry there. A proton acceptor, which is the very definition of a base. And of course, the COO over here, COOH, we know to be COOH. That's the ionizable proton there. And so this is a proton donor, which is our very definition of an acid. So a carboxylic acid and an amino group contain an acid and a base within the same structure in the amino acid. Remember the general structure, we have a carbon, and that carbon is called the alpha carbon. That carbon will have three attachments that are always the same. It will have a hydrogen, it will have a carboxylic acid, it will have a base group called an amine, and then what makes one amino acid unique from the next is what functional group is down here. This functional group gives the amino acid its identity. We'll see that there are 20 different possibilities for what this alkyl group can be and give it its identity. So the very first and simplest of all amino acids is glycine, where R is equal to H. The alkyl group is equal to a hydrogen. So notice we still have the alpha carbon. 3 of the 4 bonds are always the same. Leads out to an acid, leads out to an amine. Those groups are always the same. What gives the acid its identity, or what gives this amino acid its identity, is the identity of this R group. And we're saying the simplest of all R groups is a hydrogen. Notice that makes this carbon achiral. And it's the only 20 of the 20, glycine is the only non-chiral center. And it's non-chiral, as you can clearly see, because 2 of the same are attached to hydrogens. So remember, to be chiral, you have to have 4 unique attachments. Here there's only 3 unique. 2 of them are the same, giving hydrogens. Our group is called the side chain, and it identifies the amino acid. Now, if R is a basic nitrogen atom, the amino acid ends up to be basic. So if something down here in the terms of the R group, if R is a basic nitrogen atom, that's going to dominate the pH. Sometimes we'll see that there's an additional Cu group down there, and if it is, it's an acidic amino acid. Most of them are neutral. And most of the amino acids, when I say are neutral, it's because this R group does not contain an additional base nitrogen or an additional carboxylic acid. Just a few do, and we'll take a look at those structures here shortly. Since amino acids contain a base, which is the amine, and the acid, which is the Cu, we often see this as a proton transfer from the acid to the base, forming a Zwitter ion. You need to know that word, Zwitter. It's a fun word to say, isn't it? A Zwitter ion, all one word. Now that's what this means. Remember a moment ago I said this is the ionizing proton on a carboxylic acid, Cu. And I recognize over here that this is a lone set of electrons on that nitrogen. This is the base function when we have a lone pair of electrons. And this is the ionizing proton on that acid. So we have a proton transfer where this hydrogen gets bonded over here to the nitrogen. And what happens then is that we form an electrically neutral salt. The amine portion will always carry a positive charge, and the carboxylic acid portion will always carry a negative charge. So think about what happened. This is now the conjugate acid of the amine, and this is the conjugate base of the carboxylic acid. The Zwitter ion is how we find an amino acid in nature when it's in a pH neutral solution. The salt is always in its neutral form, where the positive ion on the nitrogen is being electrically balanced by the Cu negative. So this is the acid, and this is the base. We have a proton transfer. The acid donates its proton to the base and forms its conjugate pairs. So in nature, in a neutral solution, or relatively neutral, so there's a pH range of about 6 to 8. In a neutral range of pH, you are most likely going to find an amino acid as a Zwitter ion. You never, ever see the acid and the base form. You see the conjugate acid and the conjugate base form known as a Zwitter ion, where the proton has been transferred from the acid to the base. Let's focus in on some examples of what R is and give the identity to each of these amino acids. When I present this chart, what I'm going to ask you to do is to fill in your note pack the following area. Let me just be clear. I want to just shut this one down. I don't need it. Nope. So right now we're kind of coming down here into the 20 different amino acids. And what I'm going to say is this is an extremely important table, but I'm not going to have you memorize. Some instructors are really good at making you memorize the structure, but I'm going to ask you to memorize the 3-letter code for each of these and the 1-letter code, but the structure itself you do not have to memorize. Let me give you an example. The first of the 20 amino acids is aniline. Alanine. Hear that? I'm going to say it again. Alanine. It has a functional group where R is equal to a methyl group. That R gives it its identity. What I'm asking you to fill in here is the 3-letter code and the 1-letter code. ALA and A. And so on, whoops, I went too far. On this, back to your note pack, ALA and A. Now, back when I was a student, there were only 3-letter codes for the 20 amino acids. But now we've kind of even shortened it more, where we have an abbreviation that's 1 letter to represent the term alanine. And so, for instance, in a homework problem, you might be given, it might say, draw the amino acid that would form from alanine, and it's going to give you ALA as your structure. And you'll have to know ALA stands for alanine. So I'm letting you know that this portion of the table will be given to you as a test-taken tool, but this part of the table will not be. So for each of the structures, you need to associate the name to the 3-letter and 1-letter abbreviation. You might find flashcards to be very helpful. You'll be given the structure and the name, but you should know the 3-letter abbreviation and the 1-letter abbreviation for each of the 20 amino acids, and you'll see there's a nice trend there. So in the next section, we'll just keep going down. The next one is named asparagine. Asparagine. You can see its functional group is CH2CONH2. It has a 3-letter abbreviation of ASN and a 1-letter abbreviation of N. The R group gives it its identity. We'll need to know the abbreviations for each. Cysteine. Cysteine, let me say it again, cysteine. We have a CH2SH functional group. 3-letter code is CYS, 1-letter code, capital C. Next one I see is called glutamine. Let me say it again, glutamine. In glutamine, the R group is longer, isn't it? CH2, CH2, CO, NH2, glutamine. Abbreviated GLN, and the 1-letter is a Q. Glycine. Glycine has the most simple structure, the A chiral carbon we drew a moment ago. Its R is simply an H. GLY, G, 3-letter and 1-letter code respectively. Isoleucine. Isoleucine. Look at this. CH, parentheses, CH3, CH2, CH3. That's its R group, giving it its identity as isoleucine. This is capital ILE. ILE. I know that's hard to see in that. I, then L, then E. And then a capital I is its 1-letter abbreviation. From isoleucine, we go to just leucine. CH2CH, CH3 taken twice. LEU, L. Methionine. Methionine. I hear a sulfur in there. Methionine, CH2, CH2, S, CH3. MET, capital M. Let me say this word with you. Phenylalanine. Hear this alanine? Now this is phenylalanine. You can see with aniline, it had just a methyl group. Phenylalanine has the methyl and a phenyl. Its abbreviations, PHE, and then an F. Now that's fun, F for phenylene. Now this next one is very unique. Proline, here is the alpha carbon, and the entire structural unit here of the R group is a cyclic structure. That's very unique. So the amine portion is part of the cyclic structure. Here's the COO, C-O-O. Notice these are all written as Zwitter ions, which are really considered to be salts because we have a positive and a negative region in each. So proline is a cyclic R group, P-R-O followed by P. Serine, C-H-2-O-H. You're writing an S-E-R and S, serine. Threonine, C-H-O-H, CH3. Of course, it has T-H-R followed by T for the 3-letter, 1-letter code, respectively. Tryptophan, it has an R group that's quite extensively aromatic. A 5-membered aromatic ring as well as a 6-membered aromatic ring. TRP and W are its 3-letter and 1-letter code, respectively. Tyrosine also includes an aromatic ring, TYR, and the letter Y. Valine, VAL and V. Valine is hydrocarbon, CH, CH3 taken twice. Now we see that in this next slide, these are the naturally occurring amino acids that are acidic. That means they have an additional COO group. In the R, you have an additional COO negative. Maybe some other things as well, but you're going to see an additional carboxylic acid. So for example, aspartic acid is one of the 20 amino acids. It contains an additional carboxylic acid functional group, CH2COO negative. I want you to notice that now the structure is not electrically neutral at the pH range of around 7. Instead, it's going to have a net -1 charge. There are 2 functional groups of carboxylic acids compared to just 1 amine group. So that left it with a net -1 charge as the Zwitter ion. Here is glutamic acid. Glutamic acid has 2 methyl groups followed by a COO negative. GLU and E, glutamic acid. Now look in this column. These are the basic amino acids, meaning that the R group has an additional amine function in it. So you can see all kinds of amines. And arginine is a basic structure. Do you notice that this is no longer electrically neutral? That I have one unit here. Let me kind of highlight that better. I have an amine that's a +1, another amine that's a +1, and one and only one COO negative, telling me that I have a net charge of +1 at a pH of around 7. And that's a range depending on where this Zwitter ion is located. Here's histidine, and finally lysine. Those are the 3 basic amino acids. You notice that some of them were highlighted with an asterisk. It just means that those were essential amino acids, meaning you must intake them by diet, that your body does not make those. So you've had a chance to kind of investigate each of the 20 amino acids. Again, I'll emphasize that the structures and names will be given. Alrighty, so that part, you just have to know where to find them, and that's why I took the time to make this in your notepad. So you had a nice test-taking tool study guide to kind of put together the 3-letter code and 1-letter code for each of those 20 amino acids. Now, when we go and look at amino acids, we had mentioned that all amino acids except one has a chirality center, and that's on the alpha carbon. So for example, these are examples of Fisher projections of the amino acids. Now remember, when we wrote Fisher projections, we want all the carbon to be in the vertical line. So the vertical line contains all the carbons. And at the top, this is the carbon that has the most oxygen. And in this example, that carbon is always going to be the COO carboxylic acid functional group. So you're going to see that the COO part is listed at the top. See what I'm saying here? The COO negative is now at the top. And then whatever the R group is at the bottom. So on the vertical line, when we're drawing Fisher projections, we'll have the COO negative, and then whatever the R group is here. And then on the horizontal line, that leaves us the nitrogen group and a hydrogen group. Those are the other functional groups that all amino acids contain. And so if the amine group is on the left side of this Fisher projection, if the nitrogen is over here, that means the hydrogen must be on the right side. This is known as the L enantiomer. If the amine group is on the right side, it's called the D enantiomer. Levo and dextro rotary. Do you remember that from chirality centers? Levo means left, dextro means right. Now, something critically important to note in your notes here, the naturally occurring enantiomer is the L enantiomer, where the amine group is on the left. These do not occur in nature. That D isomer, where the amine group is on the right, is just not found that way in nature. The L amino acids are the naturally occurring amino acids. L stands for left. It stands for levo, but it means left. And D stands for dextro, which means right. The L is the naturally occurring amino acid. So these two are enantiomers. We have an L enantiomer and a D enantiomer. The L on the left side is the naturally occurring enantiomer of an amino acid. Now remember, if I were to draw glycine, glycine would have a carbon, COO negative up top. It would have a hydrogen, the nitrogen, and then this would also be hydrogen for glycine, and therefore that is not a chiral center. So therefore it makes no sense to say that it has a D or an L enantiomer. It has no enantiomers. It's not chiral. But the other 19 have a chiral center, and the way we find that in nature is based on the L conformation. Let's practice a moment. Now I know in your note pack I have drawn for you L-aniline and D-aniline. Let's go ahead and take a look at that structure and make sure that we can see what we're being asked to do. So let's scroll down a little bit and find that portion. And I'm asking us to draw the Fisher projection for the following. And I've provided one for you because it was just on our screen a moment ago. L stands for left side. Think of it that way. It doesn't. It stands for levo, but L is left. And that's the naturally occurring enantiomer. D stands for dextro, and that means right-hand side. The amine group is on the right-hand side. Another thing I'd like to encourage you to do is to draw with me the way that we find the carboxylic acid on the top as well. So let's draw in, just as we see here, the chiral center in the center of the cross, the H, and so this is alanine. Now remember alanine, just to kind of scroll back. Alanine. This takes a lot of page. Here it is, alanine. See how I'm knowing what the functional group is? It's a CH3 functional group. That functional group let me know the identity, and that's how I knew that this was alanine, because that's the R group right there, alanine. So when I write CH3 down here, I've now identified the amino acid as alanine. I put the amine group on the left for the L enantiomer. But this is the part that I want you to add on. When we take C, double bond O, I want you to notice that it's going to be about a 120-degree bond angle, and the oxygen, which is negative, is going to be attracted to the positive amine. So these guys are on the same side, the left side, that the charges align themselves to the left. Compare that to how we find the D isomer. And again, the D means the right side for dextrorotary. We have the amine, which is charged positive. The hydrogen flipped to the left side. Alanine gets its identity for the R group, being a methyl group. And now look, since the C is now in this direction, I want us to notice that the oxygen with its negative is aligning itself to the right as well, based on being attracted to the positive amine. So we can add that depth to our notes as well. Let's try another. Let's draw the Fisher projection in the L and the D enantiomer for leucine. Well, what does that mean? How do I go about starting this? Well, the first thing, I need my tool page, and I need to find leucine to identify the R group. And so this is the nice part of having this all in your notes. You're simply scrolling through until you find leucine, and now you can identify its functional group. The CH2CH, CH3 taken twice. I'm not telling you have to memorize that, but you do have to know how to find it using your test-taking tool chart of the 20 amino acids. So that's the leucine functional group. I'm going to draw the chiral center in the center of my cross. That on the L enantiomer, oops, I have the amine group protonated over here, NH3+, and now the H will be to the right. To make it leucine, do you remember how we looked up the R group? CH2, CH, CH3, taken twice. That makes it leucine. Since the carboxylic acid is COO negative, I know that we're going to align those charges to the same side, and it will be L, left side, for the naturally occurring amino acid. That is the L enantiomer Fisher projection of leucine. And of course, we just flip to do the right side. The identity comes from its functional group, CH2, CH, CH3 taken twice. This time the amine will be placed on the right side, making the hydrogen on the left side. And when we draw that carboxylic acid, the COO, we see that the charges have aligned themselves to be both on the right-hand side, that the charges are attracted to one another. So the enantiomer places the COO with the oxygen negative right close by to the amine positive. How about cysteine? How do we go about tackling cysteine? Well, if you're working ahead, you've already gone and found cysteine. We need to find its functional group. And so this skill says, in order to draw this Fisher projection, I have to be able to locate the R. Cysteine has a functional group of CH2SH. That's all I needed to know to draw. I'm going to draw a Fisher projection. On the left side for the L enantiomer, we have the protonated amine, NH3+, making the hydrogen to the right side. We said L-cysteine had a functional group of CH2SH. Whoops, I'm sorry, my hand keeps touching that keyboard. There, I got it. And then we're going to make sure that the COO negative aligns its charge so that they're both facing the left side of this enantiomer. The D, dextro, standing for right, CH2SH gives the functional group the identity of cysteine. The protonated amine, NH3+, now lies to the right, meaning that the carboxylic acid portion will also give its charge to the right, since like these opposite charges, the O negative and NH3+, attract one another. They'll align themselves to the right side. And you have one more to draw. Did you find it? This is L-phenylaniline. So we have to, again, use our chart. And the more you use it, the more you'll end up remembering these, I'm sure. Phenylaniline. Here it is on our functional group page. We needed to recognize phenylaniline as having this methyl attached to the aromatic benzene ring. So the functional group of CH2 attached to the benzene allows us to draw the Fisher projection of phenylaniline. So with this example, let's draw our Fisher projection cross. The protonated amine group, NH3+, on the left side, meaning the hydrogen is on the right. We said the functional group here for the phenylaniline has a CH2 group attached to the benzene ring. Oops, that's not very neat. And then we'll alternate double, single, double, single. Remember that the carboxylic acid will align its oxygen, the negative side, to the left side as well. So here we have the Zwitter ion of L-phenylaniline. And I'll just quickly redraw, knowing that CH2 attached to the benzene ring gives us the identity. This time we have the D, so the hydrogen is on this side, the protonated amine group, NH3+, the carboxylic acid, C double bond O, O negative. That's not very neat. You did it. We've taken the time to go through and draw each of those together. I did it on your notepad page. That gave me just a bit more room to write than what my slide did here. Now this slide is not in your notepad, but we can sure talk about it. Add it into a margin somewhere. It would be very helpful. It's just a quick thought question. It comes from your homework, so I thought I would just insert it in. It simply asks, which of these amino acids occurs naturally? And it's an opportunity to remind you that it's the L enantiomer that is naturally occurring. Think of L as left, it stands for levo, but left side works well. And so I'm just trying to recognize the amine group, the protonated amine, NH3+, has to be on the left side in order for it to be a naturally occurring amino acid. And that criteria is A and C. What if you were asked to name these amino acids? Can you do that skill? Well, that relies on just taking a moment and finding the identity of the functional group. For example, who has a functional group of CH2OH? Can you find it? CH2OH. This is a very busy slide. I had colored it earlier. But here I'm recognizing, if you can see this, serine. CH2OH, that means that molecule is serine. And so when I'm asking you the identity here, you found that molecule, oops, right here is the slide, and you identify this as serine. Now here's something interesting when I'm asked for the identity of B and C. Do you notice that both of them contain an additional acid group? COO negative. That helps me narrow down my search very quickly because they're going to be one of the two acidic amino acids. And so on my chart here for these, one of them will be aspartic and the next will be glutamic. So the glutamic acid, of course, has 2 carbons. The aspartic acid has just one carbon, and then you see the additional the carboxylic acid functional group. So when I noticed that those functional groups contained an additional COO negative, I knew that it would be classified as an acidic amino acid just based on seeing the COO negative, and that just helps narrow my search very quickly. So we labeled this one as glutamic acid, and this one was labeled as aspartic acid. Good work. So these amino acids exist as neutrally charged zwitterions at a certain range of a pH. That pH is going to be known as its isoelectric pH. That prefix iso means the same. Remember that if we see hypo, that means below. Hyper means above. If you see iso, it means the same. So the same charge throughout. What that just means is it's electrically balanced. I have a positive charge that's being neutralized by a negative charge, so the overall charge is 0. It is an isoelectric pH. You have the negative charge on the carboxylic acid being balanced by the positive charge on the ammonium cation. And this occurs at a pH close to 7. And I need your help in your notepad repairing this. Do you see where I accidentally typed a 2 in this box for you? Can you please help change that to a 6? Do you see where I'm looking? Down here, this should say pH of 6. The zwitterion of alanine overall is neutral at its isoelectric point. But that isoelectric point is at a neutral range of pH, about 6. Now, this amino acid can exist in different forms, just depending upon the pH of the environment it's in. So around pH of neutral, we know 7 is neutral. 6 is in a range of being close to neutral, as is just slightly above 7 as well. We have a zwitterion that's electrically neutral. But what if we're in an acidic range? The acidic range gives us a pH close, lower. And so where I'm looking now, if the pH here is about 2, you keep adding acid until the pH lowers. So no longer at about 6, we've added acid to lower the pH. What happens to the structure? Well, adding acid puts protons into the solution. Well, protons being protonated water molecules, right? Protons into solution. And so instead of having this negative ion on the acid, it becomes neutralized by placing a proton on it. And no longer do we have a zwitterion, but we're left with an overall +1 charge. An acid solution protonates the carboxylic acid. So it protonates the COO negative, turning it into COOH, the protonated form of the acid. So what does that do to the overall charge? Well, keep in mind here, this is still a +1 charge, but there's no other charge to neutralize it. So in an acid range, you have a +1 charge overall, where the amine is also protonated. So the amine is protonated. And the acid is protonated in an acid solution, giving us a +1 charge overall. Acids protonate both the amine and the acid portion of our amino acid. Well, what about pouring base in there? Bases, if I'm adding base, you're going to raise the pH. And I need your help here as well. pH of a neutral is 7, pH of an acid is about 2, pH of a base is about 10. So where I'm asking for an additional help right here, this should really say pH of about 10. Greater, I'm looking at about a pH of 10 for a basic range. Just help me fix that in your notes. The ammonium cation loses the proton as we're going to add base. So think about adding base. Removes the proton because you're neutralizing it. So neither one will have a proton. Notice that COOH deprotonates. This doesn't have a proton. And on this side as well, what did have a proton on it was the amine. It has been deprotonated. Bases remove protons. Acids add protons. And so now look what's happened. We have a negative charge on the carboxylic acid, but no positive to neutralize it, so we're left with a negative charge overall. So this isoelectric point is where the Zwitter ion normally predominates. Isoelectric point is abbreviated as P, and then you'll see this is an I, but it's an italicized I. So when you see that slanty, it's just the way that it gets typed with a font in most texts. That's a capital I, the isoelectric point. That's the pH at which amino acids normally exist in its neutral form, right? A Zwitter ion is what that's called. For most neutral amino acids, the Zwitter ion exists at about a pH of 6. We know that we have some few acidic amino acids. Remember those? There were a few, glutamic acid and aspartic acid. Their isoelectric points are at around 3. And there were a few basic amino acids that isoelectric points are about 7.6 to 10 in the basic range. But for the vast majority of those neutral amino acids, where the R group contains no additional functional groups of acid or base, the Zwitter ion is about pH of 6. So let's practice that. Let's draw the structure of the amino acid glycine. Now, if you haven't figured out glycine as being the most simple, oops, I spelled it wrong, moment of dyslexia. The most simplest of all, where R is just simply the hydrogen. So that's glycine. It's an achiral center, meaning that 3 of the 4 bonds, right, are always the same. One leads to a hydrogen, one leads to COO negative, one leads to the NH3 plus, and this 4th one will be the hydrogen. This is the hydrogen that comes from the R group, making it glycine. Now, at a pH of 7, about 6, about neutral range, it's a Zwitter ion, meaning that the acid is deprotonated, but the amino group is protonated. Remember how that happened? The acid donated its proton. So remember, this does not exist, but this is how it forms. The amino acid says this proton was transferred over here. And that's why we never see that particular structure in nature. It's always as the Zwitter ion, where the amine group is protonated as NH3 plus, and the carboxylic acid is deprotonated as COO negative. Now let's add acid. As you add acid, pH drops, doesn't it? Adding acid adds protons to the solution. So who would like to, remember when I say protons, just think of that as a hydrogen ion. Who would really like to accept a proton? Would NH3 plus like another hydrogen? Of course not. But the COO negative sure could. So again, as I add protons, the amine portion stays protonated as NH3+, and the acid part reprotonates to COO negative, making the net charge of this structure as a +1. Let's go the opposite way. Let's add base. As I add base, the pH begins to rise. And when you're adding base, you are going to remove protons because acids and base neutralize one another. You remove the excess hydrogen ions. Bases will neutralize the acid. And so now on this side, the acid is still deprotonated because all the protons have been removed. The amine group also goes back to a deprotonated form. NH3+ becomes NH2 neutral, making the net charge a -1 overall. The Zwitter ion is neutral. Basic solutions provide a negative charge. Acidic solutions provide a positive charge. We drew the structure of glycine at 3 different pHs. And that's how I drew that there. With glycine in a pH range of very acidic, we protonate. Both sides are protonated. Here we have a neutral solution at the Zwitter ion. The acid is deprotonated, the base is protonated. And now in a basic range, both are deprotonated. Meaning that we have a net negative charge, the acid COO negative and the base NH2 neutral. So we have a net negative charge overall. All right, we're doing great. How about pausing the video and just give yourself some think time and draw leucine in the different pH ranges. I bet you'll need a moment to go back and find leucine. What is its functional group? Pause, think time, come back. Welcome back. Did you find leucine? Well, of course, our alpha carbon, C, is going to lead up to a hydrogen and lead down to the R group. The R group for leucine you found is CH2, CH, CH3 taken twice. That R group gave the leucine its identity. Now, in a Zwitter ion, we'd have COO negative. And just to make sure you see what that really looks like, every time I take that little shortcut, don't forget that's a carbonyl carbon leading out to an O negative. COO negative. That proton was transferred to the amine group, so that's our NH3+. Therefore, this is a neutral Zwitter ion. Let's add acid. When you add acid, you're adding protons, right? When you add protons, you're going to protonate the acid portion of your amino acid. So up here is our hydrogen. Down below is the leucine functional group, CH2, CH, CH3 taken twice. Now in an acid, we'll see that the acid gets protonated. So now all of a sudden we have COOH, and the base is still protonated as NH3+. That gives it a +1 charge overall. Let's add base. When we add base to the solution, the pH rises. What we're doing is removing protons. And when we do that, all of those structures that were protonated are no longer protonated. So over here, COO negative. Let me just draw that like I did over here. That's a deprotonated acid, and the base is now deprotonated to simply be NH2 instead of NH3+. And now all of a sudden we see that our net charge is -1 in a basic range. How are we doing for time? That's a good hour of lesson. Let's pause here, take a break. We'll come back for section 4 and I'm going to record it in just a few minutes, but you can come back when ready.